attraction of electrons pulls them downhill energetically, okay? And that's what makes these reactions so potent in terms of releasing energy. The electrons fall all the way to this very electronegative material here, releasing all that energy in a blast of heat. Of course, there's an activation energy here, but this comes from the heat from a match or the other batch burning, and then this is guaranteed to happen in this exergonic energy release. And of course, in the combustion reaction, all this comes off in a blast of white heat that would kill the cell. So what a cell does, okay, and again, it's a redox reaction. The sugar is oxidized, okay, to CO2, and the oxygen is reduced to water. How do you know it's reduced? It's got the hydrogens. That's how you always know who's got the hydrogens at the end. That's the one that's reduced. Again, the electrons and the protons were grabbed to balance that negative charge. Proton is H plus, right? So you always know who is reduced and who is oxidized. Remember the sugar here, all these hydrogens here. How many hydrogens in CO2? None! So it's oxidized, right? The same overall chemistry, except now we're going to do a controlled burn. Okay? Where as the electrons tumble through the electron transport chain down a free energy gradient, so it's guaranteed to happen spontaneously, energy is released in little packages here that won't elevate the cell temperature and destroy the cell and its proteins. And the energy can be harvested in the form of a proton gradient that can be then used to make ATP. So fundamentally it's the same reaction, it's just how the energy is harvested. Here the machinery exists to harvest this in parcels. Again, it's an electron going through a transport chain down a free energy gradient. As that energy is released piece by piece, it establishes a proton gradient, which then can, like water behind a dam, is potential energy that can be used to build ATP. Now we need electron carriers, okay? As these electrons are stripped away from this organic fuel, we need a carrier of those electrons. We need a high energy carrier. And here's the most important electron carrier in aerobic respiration. It's called nicotine amide adenine dinucleotide. Nicotine amide, it's a modified nucleotide. Why isn't he here? Put it there, would you? Okay. I love their slides. So this is a modified nucleotide. Got two ribose sugars here. This diphosphate adenine here. So nicotine amide. This is the amide portion of the molecule. Those are the organic chem. This is an amide. This is the adenine ring here. Okay. I guess this is the nicotine part of the nicotine molecule. This, when it's oxidized, that means it's electron deficient and proton deficient. It's NAD plus. Okay. This electron carrier, this is what will shuttle energy electrons at high energy stripped from this sugar molecule and deliver them. This is what delivers electrons to the electron transport chain. Okay. So they can flow down that energy gradient toward oxygen to release the energy to establish a proton gradient. I'm saying a lot of things over and over just because you'll see in a second. This is our shuttle. It can receive two hydrogen atoms stripped away. As you oxidize a molecule, you strip the hydrogens away and their electrons, right? And this can store two electrons and one hydrogen proton. The other one is released. Okay. So this is the reduced form. Notice it's got you know it's reduced because it's got a hydrogen there. It's picked up two electrons, stripped away from, as we dismantle energy-rich electrons from organic fuel, and it carries. As it strips away those electrons, it also strips away the two protons, well, two hydrogen atoms, but it can only hold on to one hydrogen proton. The other one is kind of released into the fluid. So that's our important electron shuttle, NAD, nicotine amide adenine dinucleotide. Dinucleotide. There's one, there's the other. Okay. So this is an overview. And this is where we'll start on Wednesday. This chemistry here is really an overview of a lot of complexity. And here It's a summary of 3 key events that are involved in the gradual dismantling of organic fuel to extract the energy in a usable form in a controlled burn. We have glycolysis, which literally means sugar splitting. That's the first part of this energy harvesting pathway. This is, we say, the anaerobic portion. This requires no oxygen. This metabolic pathway, 10 reactions here, probably the oldest metabolic pathway in the history of the planet. This has been around for billions of years. Everything does glycolysis, everything. Whether you're aerobic, anaerobic, bacterium, prokaryote, eukaryote, doesn't matter. Everybody has the enzymes to do this. This is the key foundational metabolic pathway of life. It's the original way living cells could extract high energy electrons from sugars to make ATP, but the yield was crap. For every sugar molecule, you only could make 2 ATP net yield. It's something, but it's not a lot. Okay? It happens in the cytoplasm. No fancy organelles required, because it precedes the evolution of organelles. It's more ancient than the mitochondria. It splits a sugar, splits a 6-carbon sugar into 2 3-carbon sugars. They're called pyruvates or pyruvic acid. And those are imported into the mitochondria. Okay? On the way in, they're going to lose a carbon to become what's called an acetyl group. I'm going to go through all this in detail. This is the overview. And then they're bound to a modified nucleotide, sort of like a chaperone. Like in my first date, my mother was a chaperone. Boy, that was a lot of fun. And the 2-carbon remnants of this split sugar are going to then get dismantled through a series of chemical reactions that form a cycle. What that means is that the accepting molecule here gets regenerated. That's why it's a cycle. It's called the Krebs cycle, after Hans Krebs, who described it. Okay? The citric acid cycle, based on the first product of the reaction. This cycle in the matrix of the mitochondria completely dismantles the remnants of the sugar split in half. So at the end of the Krebs cycle, all that remains of the sugars are CO2s. And how many CO2 is liberated from the mitochondria at the end of respiration? 6. You have 6 carbons of the sugar. Okay? And they just diffuse out. Okay? In this process of dismantling the remnants of the sugars that were, when it was split in half, the main part of this is to extract the high energy electrons onto shuttles like NADH and another shuttle. The electrons are stripped away, kept at high energy. A little ATP is made, but not much. And then the big payoff in terms of making ATP is the electrons stripped from these sugar fragments, and the sugar at the end is gone back to CO2 back in the world. Those electrons on these high energy shuttles like NADH delivered to an electron transport chain, which in the inner membrane here, the series of proteins bound, complexes bound in the membrane, the electrons flow energetically downhill toward oxygen. As they do so, they generate a mountain of ATP. Your yield goes, in the original glycolysis chemistry, your net yield was 2 ATP. At the end of this, you're up to like 32. You've got a 15-fold increase in energy harvest. And now you can be a much more active, energetic animal. Okay? And it's this last step that requires oxygen. The electrons flow through the membrane, to the transport chain, toward oxygen. So we're going to look at these in detail. We'll look at the events of glycolysis, the ancient anaerobic pathway, and then with oxygen, how the degradation products enter into the mitochondrial matrix where they're stripped of their electrons in the Krebs cycle. And those extracted electrons on shuttles like NADH delivered to the inner membrane, the electron transport chain, to build a proton gradient, like we saw on the very first slide, which powers ATP synthesis. That's probably a good place to stop. Let me ask you one thing here, because tomorrow's our lab exam, 1. Now I'm going to guess that some of you have access to my lab exam 1 from last spring. I'm sure that somebody knows somebody and they've got a copy of my old test. I'm not even going to ask anybody to admit that. I'm just going to assume that's true. I know I was a student. You always look for the old test, right? So what I'm going to do, now I would have done this over the weekend, but I didn't have power to my house till yesterday. I didn't get home till Friday, and I had no power till yesterday. So that's why you didn't get the emails from me about the data until yesterday. Okay. So what I'm going to do is I'm going to post on Canvas when I get home tonight the old test. Okay. So you can look at what the old I don't have time to put the answer key together, but you should see what the old test was like. Okay. And tomorrow's lab exam is based on that test. Okay, because I want you to redo it. Okay. All right, we'll see you mañana.